Okay, and then I have the pleasure to introduce Dr. Agata, I hope I say your name correctly, Dauskowska-Golek.
Perfect. Let's.
So a good friend of mine, and she's an associate professor in biology at University of Silesia in Katowice, and the leader of the Plant Genetics and Functional Genomics Group. And since 2019, she has been the Deputy Dean for Science Promotion, which is very important, and internalization of the Faculty of Natural Sciences. So her research is more focused on the molecular basis of plants' response to a biotic stress adaptation. She has been very successful in getting international and national funding through several projects, including participation on the famous Barista project under the frame of Horizon 2020. And she's now working together with me and others that are in this call on the Recobar project. And today she was talking about functional genomics research taken on barley, ABA-related mutants. So Agata, the floor is yours.
Thank you very much, Sonia, for such nice introduction. I will share my screen with you. Just please let me know if it's visible. The slides are visible.
Yeah, we can see the slides. And for questions, please put them in the chat, and in the end, we'll go through them.
Okay, thank you very much one more time for the nice introduction. And also I'd like to thank you for invitation, for giving this talk, for sharing our research with you. I'm really glad to do it, and yeah, excited also because it's always excitement behind science. So let me start with the outline of the talk. Mostly I will focus on barley mutants in ABA regulators with better drought performance. Then I will tell you briefly about other subjects of our study in our group, in plant genomics group, which is also focused on brasinosteroids, metabolism and signaling, some strigolactone story, and also root hair development investigation using barley mutants. Then a little bit of aluminum toxicity, which is a really emerging topic in our group and nicely covered also by research of one of our members and her students. So let me start with drought stress. As all we know, it's really a great threat, and taking into account arable land affected by drought stress has increased twice in the last 40 years, and 80% of economical losses in developing countries caused by drought affected already the agricultural sector, and it's still increasing. The drought stress has risen by 30% since 2000. We really need to do something. And as plant scientists, in the face of climate changes, we can identify, for example, regulatory genes which are key in drought response pathway. Then we can discern the mechanism of drought response, also use that knowledge finally to control plants' drought response. But above all of that, we should act together. And there is a lot of initiatives that can be taken and are taken by plant scientists. One of them is Plant Act, which I strongly encourage you to check. I have a great pleasure to be a member of that. The idea came to our minds during the Montpellier symposium last year. It was symposium of IMBO Society focused on plants, molecular basis of plants' response to climate changes. And if you can scan this QR code, you will know much more about Plant Act, and there is still space for any eager to join scientists, plant scientists mostly, but we are also looking for contacts to stakeholders, because as we all know, it's really important to engage stakeholders, dissidents, and other which are really important to act together. We published in February this year in Trends in Plant Science a white report where we focus on main things that we can start with to change actions against, to fight climate changes as plant scientists. But going to drought stress, we are using mostly tilling population as a source of mutations, as a source of induced mutations. And Hortilus is a platform, a tilling platform, which we developed in our group several years ago and still maintaining it. So when we did a proof of concept study for 32 genes, which we tilled at the beginning of our journey, of our adventure, we checked the density of mutation, and for these 32 genes, we were able to obtain 1 per 477 kb mutation density, which was really high in terms of barley tilling populations. So today, in that part which is focused to drought stress, I will tell you a story of one of these 3 mutants on ERA1B. I will focus on one of them, but in terms of drought stress, we have several mutants. One of them is CBP20, the second one ABI5, and the third one ERA1, as I mentioned. We characterize them in details in case of physiological and molecular response. And for example, in case of CBP20, we noticed that it's able to deposit more epicuticular waxes at epidermis of the leaf. It's closing its stomata much faster, and also the molecular mechanism which is standing behind the drought response is focused on alternative splicing. In case of ABI5, which is transcription factor, one of positive regulators of ABA response, we noticed their massive activation and repression of stress-related genes. In case of ERA1, ERA1 is one of the gene encoding the subunit of farnesyl transferase, which is the enzyme that I will tell you more about today. And we've noticed that farnesylation, which is performed by ERA1, is a negative regulator of photosynthesis in terms of drought stress. So let me tell you a brief story about farnesylation in terms of drought stress in barley. Farnesylation is post-translational modification of the protein, which is basically the addition of prenyl group to the end of the protein. The prenyl group is added to highly conserved motif, where the cysteine is playing a key role. And the farnesyl transferase, the mutants in farnesyl transferase genes in Arabidopsis are hypersensitive to abscisic acid during seed germination, and also are tolerant to drought stress in Arabidopsis, in wheat, and recently also in rice. So several years ago, we asked the question, can we obtain drought-tolerant barley with mutation in ERA1? And then what is the mechanism of ERA1 action under the stress? So as I said, farnesylation is performed by farnesyl transferase, which is built from of 2 subunits, alpha and beta. Beta is green one on this nice GIF. And this subunit, the beta subunit, is encoded by ERA1 gene. So we tilled this gene and identified plenty of mutations, 14 mutations. Among them were 2, the most interesting to us, missense mutations, 1B and 1D. And these 2 mutations occurred in the domain which is encoding farnesyl transferase catalytic domain, so very important for the farnesylation. And as you can notice, both of them are substitution of glycine in very conserved position. So the most interesting is that this position is conserved across the species, not only plant species, because ERA1 is also highly conserved across the species, the animal species also. So when we looked at the phenotype of these mutants, we didn't observe any massive changes in the phenotype at the seedling stage, and then when we did post-harvesting analysis also. The one thing that we observed was decreased number of seed per plant and also decreased total seed weight per plant in both these mutants, in ERA1B and ERA1D. But when we looked at the drought performance. And because I'm a great fan of translational genomics and I take advantage from Arabidopsis also, I did my PhD on Arabidopsis. So I have a sentiment to Arabidopsis. I used also in our research, we are using also Arabidopsis mutants. And we also used in this research mutant, an insertional mutant of Arabidopsis in ERA1 gene. So we've noticed that both barley and Arabidopsis mutant performed better under drought stress. They had higher relative water content in leaves. And also what was very interesting, they didn't perform worse in case of photosynthesis under drought stress as the wild type. So you can simply notice that, for example, in the case of the wild type in Arabidopsis, there is a huge decrease in antenna size of the photosynthetic antenna, whereas in the mutant, the decrease is much more slightly decreased. Then when we looked at reaction centers, which is really crucial for photosynthesis performance, we also observed the decrease in the wild type, but it was not changed in the mutant. The same was about performance index, which is the complex parameter which express the performance of photosynthesis. And it was the same in the case of barley, whereas in the wild type we observed decrease in a lot of parameters related to photosynthesis. We didn't observe any decrease in case of the mutant. So then we asked the question about the molecular mechanism behind it. And for this purpose, under drought conditions, we performed transcriptomic analysis. So you can see the protocol here, the scheme of the protocol of drought treatment. So plants were growing in optimal water conditions. Then we were decreasing the soil moisture and drought occurred for 10 days. Then we were increasing the soil moisture to get the recovery phase. And when we performed this study only on ERA1B mutant, it was the first identified and characterized in terms of drought stress. We've noticed that after drought stress, there was a massive upregulation of genes that could be classified in biological processes related to galactolipid synthesis. And galactolipids are special lipids which are building the membranes, exactly the chloroplast membranes. So we thought there must be something between farnesylation and photosynthesis protection in case of barley mutants in ERA1B under drought stress. So now we performed sequencing of transcriptome for both of these mutants, for ERA1B and ERA1D. And we performed it, of course, in control conditions and also after drought stress. And when we filtered out the genes which were common for both of these mutants, for ERA1B and ERA1D, we identified 475 genes which were dependent on mutation, on ERA1B mutation under drought stress. And among these genes, gene ontology said that there is also a lot of genes involved in lipid metabolism, which was the first hint when we performed microarray study several years ago. So we looked closer at these genes, and what we've noticed, there were some related also to synthesis of major structural component of photosynthetic membranes, in chloroplast, of course. So there was another hint that when we focus on this pathway related to galactolipid synthesis and lipid synthesis, we connect it with better photosynthesis. Then we ask the question, okay, what is the content of galactolipids in the mutant and in the wild type? And what we noticed was that after drought stress in ERA1B mutant, there was increase in the content of galactolipids, the general galactolipids content in the mutant after drought stress. So it led us to another question. We asked a question, what about ultrastructure of chloroplast in this mutant? So we performed ultrastructure analysis, and what we noticed was that already in the control conditions, the stacks of thylakoids were much more higher in mutants than in the wild type in the same conditions. And after drought stress, they were less distracted when compared with the wild type. So we counted several parameters, several parameters in these images, based on these images, and we noticed that these structures, the black structures are plastoglobules, and the number and the size of plastoglobules are different in the wild type and in the mutant. So when you look at the size of plastoglobules, you can notice that after drought stress in Sebastian, which is the wild type for these mutants, the size of plastoglobules is increased really massively, whereas in the mutants increased like threefold, twofold. And then when we looked at the number of plastoglobules, the number increased only in the mutant, but in the mutants, in both mutants, but not in the wild type. So we ask a question, a bigger plastoglobule, sorry, it was too fast. The bigger plastoglobules in the wild type, they are friend or foe? And why so many plastoglobules and smaller in size in the mutants? So going further, we ask another question. Maybe these plastoglobules are important for the photosystem repairment after drought stress, and that's why mutants behaved better. But to check it, we had to perform stress treatment in further stages also to see if the recovery is faster or not, and what about recovery in general in these mutants. So we performed a drought stress analysis in plant array system at the heading stage, and we noticed almost the same tendency of better photosynthesis also in older plants in further stage of development. And what was really interesting, we noticed also that the mutant was able to recover much more faster than the wild type when it comes to daily transpiration level. So already at the first day of recovery, the wild type was like 62% of the control, whereas the mutant was already 88%. So it was much higher daily transpiration, almost at the level of control before drought treatment. So we hypothesized that the recovery is faster in the mutant because of better photosynthesis performance. And what we suspect was the use of lipids, which are stored in plastoglobules. So we perform ultrastructure analysis of ultrastructure of chloroplast also after recovery at the seedling stage this time. And what we noticed was again differences between genotypes. So let me remind you, after drought stress, there was increase of size of plastoglobules much more profound in the wild type, and in the mutant less. And then the number of plastoglobules was increased only in the mutants. So after recovery, we've noticed that there was a decrease in case of the wild type and a slight decrease in the case of the mutants when it comes to the size of plastoglobules. But when it comes to the number of plastoglobules, it was decreased only in the mutants. So we thought that probably mutants use plastoglobules more efficiently as a source for photosynthesis repairment. So when we had a better photosynthesis, also at the stage of, also at the heading stage, and the barley is a real plant, not like Arabidopsis, we always look at the yield, at the water use efficiency. So we checked it also in our mutant. And at the plant array system, we've noticed higher water use efficiency in the mutant. In ERA1B mutant, it was tested on the platform. And when it came to the yield, we've noticed that the seed number per plant and the total grain weight per plant was not changed in the mutant when we compared control and the drought. Control and the drought. But when we compared the wild type seed number and total grain weight, we noticed decreased expected decrease after drought stress. So then we checked if ERA1 mutation in different genetic background, for example, the elite cultivar background, will behave the same. So we introgressed ERA1B mutation in the background of elite cultivar. In this case, we introgressed it in plant RGT. You can see on the scheme how we did it. And at the end, we finished with 4 lines with introgressed ERA1B mutation in RGT plant background. And together with our collaborators in Barista project, with Ana Casas and Ernesto Igartua, we genotyped them using 50K iSelect SNP array, which all of you know perfectly. And all these 4 lines were like above 90% shared monomorphic loci between introgressed line and RGT planet, whereas Sebastian and RGT planet shared almost 80% of monomorphic loci. So this result was really promising for further studies. And we studied also response of these introgression lines under stress, under drought stress, for example, in plant array system. We checked water use efficiency based on photosynthetic rate and stomatoconductance, and ERA1B introgressed in RGT planet exhibited higher water use efficiency, similarly to original T-link mutants under drought at flowering stage of development. Then we performed the first study in the field condition for these lines, for these introgression lines in RGT planet background. And what we noticed was that all of them, except for one, for DH1 is that line, performed better in terms of photosynthesis under field conditions, under drought conditions in the field. And only this one, which is worst, this DH1 line, seemed to activate photoprotective mechanism exactly as planet did. So only these 2 displayed increased anthocyanins level. So it was really interesting observation. And then we looked also on several parameters related to yield. So for example, in case of total grain and weight of total grain, we didn't notice any worse behavior of introgression lines when compared with the planet RGT background. Then going back to the molecular analysis, we hunted also for potential targets of ERA1 farnesylation among differentially expressed genes. So in this study, we used 2,500 barley proteins encoded by differentially expressed genes, which were identified in this first study based on microarrays, which I told you at the beginning of my speech, for farnesylated motifs. So we identified 16 proteins with these highly conserved motifs that can be potentially recognized by ERA1, and they are listed here and were published already. But now we are looking for another potential targets using RNA-seq data that I presented you on previous slides. So because ERA1 is somehow involved in abscisic acid signaling pathway, we also were very curious about its response to exogenously applied abscisic acid. So this time, together with our Estonian partners, also in famous Barista project, we checked stomatoconductance in VPD conditions. What we noticed was that wild type, in response to ABA, closed its stomata really fast, whereas in the case of the mutant, there is no impact on stomata closure in case of ERA1 gene mutation. And what is more, another observation was that ERA1B mutant already in the control conditions exhibited lower stomatal conductance than the wild type. So then we looked at the literature because the result was really striking to us and surprising. And it was the same case in case of Arabidopsis era mutants published already by Yalakas several years ago in Hannes Colley's lab. So it was highly conserved and universal response of these mutants in different species, which was really amazing discovery to us. So we looked closer at the transcriptomic level and noticed that in genes which were upregulated already in the control in the mutant and then after abscisic acid treatment, the vast of the genes were related to chloroplast and photosynthesis. So there should be really something about this photosynthesis and abscisic acid related pathway also in the mutant. When we looked at the genes that were highly upregulated and then slightly downregulated after ABA treatment in the mutant, the most of the genes were involved in peptide biosynthesis and RNA metabolism biological processes, which was also interesting in terms of the role of farnesylation. So summing up this part of my talk, both mutants of era 1 in barley and also the insertional mutants in Arabidopsis were tested under drought conditions. And what we noticed was better photosynthesis and water-saving mechanism in era 1 mutants, which was displayed by better photosynthesis under drought, less touched by drought chloroplast ultrastructure, higher content of galactolipids, and this enigma of plastoglobules that has to be discerned more. It was observed at the seedling stage of development. Then we noticed at further stages of development faster recovery of the mutants, of era 1 mutants at the heading stage of development, and also better photosynthesis and higher water usage efficiency. There was no negative effect on yield when exposed to drought at field and control conditions, also in allied background, but here I would like to underline that we are very cautious about this result because as all of us know, this need to be investigated in more seasons and more replicates. So this is something that we are checking right now. Also, and the lower somatoconductance under optimal conditions, it's really interesting also in terms of further exposition to drought stress. Era 1 seems to be not impact on stomata closure in response to exogenously applied abscisic acid. And one of the most important statement also is that there is an evolutionary context of era 1 action as the response to drought stress seems to be similar across the species in barley and Arabidopsis as the example. So now I would like to briefly tell you and touch some other aspects that we are studying in our group. And the first one and the next one will be related also to drought somehow, but more focused in the first case on brasinosteroids metabolism. This is the study of Damian Gruszka, who is the expert in the field of brasinosteroids in our group. He's working mostly with nearly isogenic lines in Bowman cultivar. And here you see, for example, several mutants. The blue are metabolism, mostly biosynthesis mutants, and the red are perception mutants. So both of them are in brasinosteroids receptor. This one is USO, famous allele in this gene. So all of them, mutants, in brasinosteroids mutants, were better in terms of drought stress when compared with the Bowman cultivar. All of them had higher water content in the leaves when compared with the Bowman. Next, what is also interesting is rubisco activity under control and drought conditions. As you can notice, it was already lower in the mutants, in the control conditions, in the mutants, all of them, and decreased by 40% after drought stress, only in Bowman, whereas in the mutants, it stayed at the same level almost as it was in control conditions. And this this effect was independent on genetic backgrounds of the analyzed mutants, since the same observation was made for semi-dwarf mutants in the Lisa cultivar, which we also have mutants in Brazilian steroid pathway identified among all by Damian. So then really interesting observation also regarding accumulation of endogenous Brazilian steroids in response to drought stress. So we have a castasterone, which is the biologically active form of Brazilian steroids in monocots. And as we can notice, after drought stress, the castasterone content is increasing in all of the mutants, but the most profoundly, it was observed to be increased in Brazilian steroid insensitive lines, so in these perception mutants. It's clearly also based on research that castasterone regulates the physiological reaction of barley plants to drought. And then there is another interesting thing, because we have also 28 homocastasterone, which is biologically inactive form of Brazilian steroids. And when this form of Brazilian steroids is demethylated, it starts to be active. It starts to be castasterone. So when you look at this part of the graph where the 28 homocastasterone is measured in control conditions, and then you come back to castasterone, you can clearly notice that the accumulation of this inactive form of Brazilian steroids is exactly inverse when compared with castasterone accumulation. And it's not increasing in drought response. Even you can notice that it's slightly decreasing, which is not statistically significant, but it's slightly decreasing. And we suspect that it's just used for pumping up the form of active Brazilian steroids in mutants, in Brazilian steroid-related mutants. So the next thing is strigolactone, hormones, and here Marek Marzec is the expert in our group of strigolactone mutants. So at first, in 2016, he published his first paper regarding strigolactone's mutant, and D14 was the first one identified and characterized in our lab. It's the mutant in the receptor of strigolactones, and now we have a, let's say, collection of strigolactone mutants in our lab, identified using T-Link, of course. And we investigated further the response to drought stress of D14. And what we noticed was that strigolactone insensitive mutant, insensitive D14 mutant, was more sensitive to drought stress. It was intolerance. The drought stress was really harmful for these mutants, and these mutants exhibited lower leaf relative water content, impaired photosynthesis, disorganized chloroplast ultrastructure, altered stomata density, slower closure of stomata in response to drought stress. And what was really interesting to us, it was again evolutionary conserved traits. Most of them we observed also in D14 mutant in Arabidopsis. So then we performed multi-omic approach. What we mean behind that is the hormonomics, the proteomics and transcriptomics. So here I'm showing you just abscisic acid and phasic acid because they are the most profound effect in terms of the sensitivity to drought stress of this mutant. So as you notice, and it's normal expected situation, the drought evoked abscisic acid synthesis. In the wild type, the abscisic acid increased, but what happened in the D14 mutant, it's increasing massively, but the mutant doesn't really feel it. because of its insensitivity to abscisic acid. So then we performed transcriptomics and proteomics, and here you see downregulated and upregulated genes, here proteins, but we wanted to get as much as we can knowledge from these omics analysis approaches. So we looked at genes that were differentially regulated at the gene expression, but also at the protein level, because as we all know, it's really hard to correlate transcriptome and proteome response. It's not 0 to 1 because of many steps between these two expression phases. So we identified 151 common DEX and DAPs in D14, and most of them, when we performed GO enrichment analysis, were involved in very basic adaptational processes to drought response. So loss of strigolactone signaling directly seems to impact multiple core adaptive drought responses, and does so at both at transcriptome and also proteome level. So it was really interesting to us, and now we are waiting just for the outcomes from reviewers, because it was submitted recently to the journal, so we will see what the outcome will be. In our lab, we are working for a long time on root hair development also in barley, and the collection of root hair mutants are developed, and the mutants are identified and so on mostly by Beata Chmielewska and Agnieszka Janiak in our lab. So we have plenty of them identified. You can look at them on this in this table, but here I briefly tell you about one of them identified using T-link RTH3 mutant, which is identified in a gene which is copper-like protein identified in maize and really well characterized in the lab of Michael Holdinger in 2008 in maize. This gene seems to play a really key role in root hair formation in barley also. It affects not only a root hair elongation, but also differentiation of root epidermal cells. We identified among all also a stop mutation mutant, which is that one RTH3H, which exhibits primordia that undergo little elongation and abnormal morphology. So as you see at the protein level, the protein is shorter, of course, because of the stop mutation. The mutation co-segregates with the analyzed mutation, with the phenotype of these mutants. And here is the phenotype of this mutant. So because we are really focused on drought stress, we also checked its drought response. And we noticed no differences at the physiological level under drought when compared with the wild type in these basic parameters that you can see on the slide. So I mean relative water content, stomata conductance, chlorophyll content, anthocyanins content, and also flavonol content. But of course, we are investigating it in more depth, looking for other traits to see if there is any differences in any other level. And the last thing I would like to mention is aluminum toxicity, which evoked DNA damage repair pathway. So this is mostly Miriam Szurma-Zubrzycka project, funded also under Horizon 2020. It was a CAPS project. And there we focus on aluminum, which is the third most abundant element in Earth's crust. And when the soil is acidic, these ions of aluminum are really available for the plant and cause root growth inhibition. So the threatening thing is that more than 50% of world's arable lands are considered to be acidic. So the aluminum toxicity is another abiotic stress that should be really that we should be really concerned about. And what is interesting also, aluminum is also a genotoxic agent, and it induces DNA damage repair pathway. So from this basic, let's say, basic scheme of DNA damage repair pathway, we can identify ATR as the master regulator, and here we focused. So we wanted to check if the mutant in ATR gene in barley will perform, how it will be performing under aluminum stress, and what about its DNA damage repair pathway. So we identified a mutant using chilling. This ATRG mutant carried missense mutation, but not in the kinase domain, but before it. And what we noticed was the root phenotype. Already in the control conditions, we observed shorter root, and it was not further reduced in response to aluminum, even in really high dosage. Whereas in the wild type, the root was decreasing its length, accordingly the concentration of the aluminum. The seminal root diameter is another indicator of aluminum stress. So basically the root diameter is increasing in response to aluminum, which was observed in the wild type. And in the mutant, the seminal root diameter was already bigger in the control conditions and didn't change after application of this concentration of aluminum. The most surprising was that the above ground part of the plant were not changed at all when we compare it with the wild type. The plant height, the spike length, the onslaught, the number of spikes were almost the same, with no statistical differences between the mutant and the wild type. So ATRG mutants exhibited reduction of root length already in the control conditions and didn't reduce this length further in response to increasing concentrations of aluminum. So then we looked at DNA damage and the cell cycle. So you can compare pictures for Sebastian for the wild type and for the mutant. So here is the DAPI staining and FIT staining. On these pictures, you can easily discern the nuclei which contain DNA which is damaged. So you can also observe that already in the control conditions in the mutants, there is much more DNA damaged nuclei than in the wild type. And it's not changing when increased concentration of aluminum is applied, whereas in the wild type, there is increase of DNA damage, which is even better visible when you look at the tunnel test outcome. You can see that in the mutant, there is a high content of nuclei containing DNA damage, and it's slightly increasing in response to aluminum, whereas in the wild type, it's increasing with increased concentration of aluminum, but at the beginning, there is no DNA damage almost. So the mitotic index is also something that we measured, and as expected in relation to this result, we observed decrease in mitotic index in Sebastian in the wild type, whereas in the mutant, there was no significant changes at all when it comes to mitotic index. So we ask a question, okay, at what phase of mitosis of the cell cycle the problem exists. So we perform cytometer analysis, and what is also interesting, there was an increase of cells containing cells that were in G2M phase of cell cycle. So it means that the arrest of cell cycle occurred here in G2M phase of development. As you can notice also in the mutant, there was no statistically differences between G1 and S phase, When it comes to G2M, it was slightly increased. In control conditions, sorry, in the wild type and in the mutant, there were no changes at all when compared all of these three phases. So what we concluded from this research was that reduction in root growth in response to aluminum is at least in part, of course, because the whole process is much more complex, but at least in part triggered by the ATR-dependent activation of DNA damage repair response, leading to the arrest of the cell cycle. And I could tell you more and more about our research of our team, plant genetics and functional genomics team, but I think that touching all these aspects for now is okay, and I'll be happy to answer your question. Here are some people that I would like to acknowledge. So these are people from our team, from plant genetics and functional genomics group. Here are people involved from the Institute of Biology, Biotechnology and Environmental Protection, and I'm also acknowledge a lot of our collaborators, which we really are glad to collaborate. Here are our funds, and here is a little helper, my son, who is often with us in the greenhouse when he always enjoy staying with us. And thank you very much for your attention. And as I said, I would be really, really happy to answer your questions.
Thank you so much. It was excellent. Thank you. And yeah, really, really fantastic talk. So there are a couple of questions on the chat. So first of all, Robbie apologized, but he had to go to a different meeting. But he said that it was a very good talk and a great demonstration and value of barley mutants to explore fundamental biological processes with very nice data. So well done, really nice. So I'll start you with a first question. From Laurie Brown. Not sure, did you see any difference in the root systems of the ERA1 mutants?
Thank you very much for this question. We are investigating it right now, honestly, because we were really focused on the above ground part of the plants. We were really focused on this chloroplast related story. So we wanted to make it as deep as it's possible to discern this pathway. And recently we performed the experiment where we investigated also root of ERA1 mutants, but the data are so preliminary that I would like not to share yet, but it seems to be really interesting. And there is also interesting story behind that, because we've also observed some genes related to root development, which were in transcriptomic data, so in ERA1 mutants. So it might be really interesting. So yes, we are looking at these traits right now. Thank you very much. And when I get some results enough interesting to be shared, I will for sure.
Thanks. And then another question from Robbie. He just asked, I probably won't be here to ask this, but given they appear to provide an advantage, are non-functional ERA mutants observed in genotypes originating from natural drought-prone environments?
Yes, so this is also in progress. I've corresponded with Robbie about it some time ago, and together with Miriam Schreiber. I don't know if Miriam is with us.
I think she was here. I think I saw her.
Yes. So Miriam was so kind to perform this analysis based on population of land races and cultivars for looking for SNPs in ERA1, also in ERA1, but also in other ABA-related mutants, so genes. So we are looking at these changes, and there is something interesting. There are interesting haplotypes in some land races, but I think it's too early also to say something really about it.
Good.
So we are looking at that. Yes.
That's very good. Then the next question from Amit Kumar, how to predict start of heading date? And were you on Golden Promise? Any idea about this?
Yes, so about Golden Promise, I do not have really huge experience, but regarding the heading stage, based on the scale of barley development, the heading stage is when the half of the spike is visible. Please correct me if I'm wrong, but I think this is the one way to identify this date of heading.
Yeah, so normally it depends. So there is a paper actually distinguishing the heading date and the
Yes.
flowering
Yes.
time. So when the tip of the on is like 1 inch in, that's when you would say that flowering is occurring. Uh, so I'm not sure if anyone else wants to comment, or even Amit, if you want to comment.
Yeah, actually, I
Mhm.
have a more question about this. Any kind of gene or marker by which we can predict the start of heading date? Because I am working on cereals, rice and barley. For rice, I can easily predict, but for barley, sometimes if we are growing plants, there are number of plants. So each of plant have different date of opening of head. So it is not 100% sure for every plant that this is the start of heading date. So maybe any kind of marker.
Not sure. Do you know anything about it? No.
Really.
No.
Sorry.
Yeah.
So when we are working with the specific genotype, we are looking at the 50% of plants have this stage of development. So we say, okay, this is that stage that we want to apply any stress or any other thing. But when it comes to markers, genes, I do not have idea about marker genes. Maybe somebody from
Maybe
the
someone else in the community or in the call can reach out and contact you. Yeah, we do the same. We just go 50% of the plot
Yeah.
on that stage. It's heading date and yeah. Um, so the next question, also from Amit, better system to study effect of drought on barley roots?
Yeah, it's a hard question. And I think that we deal with this problem all because it's really hard to study roots under drought stress. And honestly, we will deal with that in Ricobar project, where we wanted to study roots of selected genotypes under drought stress. So yeah, the one idea is, and it's depending on the soil where you perform this treatment, because the easiest way is to take this plant from the soil and to make a scan of roots using WinRhizo, for example, software. But you need to be really cautious about it because it's really easy to destroy roots. So then when you scan roots, you need to make it really, really clean to get rid of all the soil and other things in that. So it's really hard to make it in really detailed way. The easiest way is just to take the root from the soil and measure its length, for example. But it's the basic one. So we are talking about it a lot now, also in our
Yeah.
team, because we want to
Yeah.
investigate it under drought conditions in this
Have you ever
recover
tried
project.
rhizotrons? Did you ever try it, or even building in-house DIY rhizotrons?
Yeah, so something like rhizotrons, let's say, or you can use these long tubes where you grow
Yeah, yeah, yeah.
plants. So we used it a lot, but the question is also what you put inside. If you put inside, for example, the sand, it would be much easier to take these roots and measure, but the sand is not a real ground where the barley is naturally living. So it's always tricky to measure
Yeah.
barley roots under drought
The
stress.
Eden half. Yeah.
Yeah.
It's very hard. And even to take core from the field's core soils, it's harder because
Yes.
you just have to have the equipment
Can I
with the
can
strength to
can I
penetrate. Yeah.
ask more about this? So, as I read in many publications, people generally use peg-based hydroponic media.
Um,
Yeah, but
So,
yeah.
PEG is not.
but
Yeah.
yes, yes. So I understood from papers and from from previous experience, peg-based drought stress are not similar type of stress as compared to soil system. But in my
Exactly.
case, my candidate gene is have a more role in root growth and have role in drought. So I have to predict the targets of my candidate in root system. So in my case, it is very urgent for set up hydroponic like things.
Yeah. Yeah.
Mm.
Yeah, it depends on the research question. What's the best phenotyping method for each person, each research question? Yeah.
Yeah.
It's not easy and it's not straightforward. Yeah. So, sorry, we'll have to go through to other comments. So from Luke, because we are on the time, so for timekeeping, um, does the ATR mutant show a meiotic phenotype?
Yeah, I expected this question from Luke. So, Luke, unfortunately, we didn't check any meiosis related traits in ATR. But I think that I will inspire Miriam, who is in charge of this research, to check it. If she's interested and perform this research, I will share you the output. But no, we didn't perform this meiotic phenotype in ATR mutants.
And the next question from Laura. Uh, Thank you very much for a nice and comprehensive talk. I know it was probably not your focus, but did you notice any differences in disease susceptibility in your field trials? Just very interesting. Smiley face.
Yeah, thank you very much, Laura, for this question. Honestly, I didn't notice it. Maybe something more can tell Anna or Alejandra, if Alejandra is still with us from CSIC, because they performed these field trials for us of these ERA1 mutants in Zaragoza in Barista project, but I think that it was not the focus. So maybe just the observation, you know, when you are going through the field and see something, but there were no scored traits regarding that. But ERA1 in Arabidopsis was already mentioned in case of pathogens. So if anybody is interested in that, I'm really open for collaboration in that because I'm not an expert in pathogen. We did not notice any difference, Anna said just on the
Yeah.
chat. So, okay, so it was not scored as I thought. So the focus was drought stress response. So it was the main aspect. But as Arabidopsis ERA1 was also published in case of pathogen as is in case of pathogens, it would be really interesting also to us to study it. So if anybody is interested in our community, I'm open for collaboration.
Yeah, because especially because Spain could be quite dry, and it was in drought in a very dry condition. So probably in a more humid environment where you have more fungal pathogens, could be interesting to look at it. Yeah. So I think we are done. Oh, so sorry, just a final question because we really are on the time clock. So from the University of Nottingham, Ricardo, did you ever thought about soil non-destructive X-ray CT scanning? You can also test root keto branching in this system since your ERA1 mutants are ABA insensitive. Great talk.
Thank you, Ricardo. Honestly, when I read this paper about xerobranching, I was really excited and thought that I have to write to you, to Malcolm, about this because, yeah, this is something which is really interesting in case of this mutant also. And this insensitivity to ABA is not 0 to 1 trait also in the case of these mutants, because when it comes to seed germination, it's sensitive to ABA, and when it comes to stomata conductance, it seems to be insensitive, so it's more complex. We cannot say that this mutant is sensitive or insensitive because it's dependent on the stage in case of this plant. But yeah, if there is possibility to use this X-ray tomography, it would be great opportunity to test this mutant. So I will email you if I can and discuss it more. Thank you very much.
Yeah. Well, I think there's not just from Laura again. It's very interesting, and thank you very much. And good to know that we have these mutants. So thank you so much, everyone, and thank you again, Agatha, for accepting the invitation from from us and to give this excellent talk. It was fantastic. Thank you so much, and thank you all. And we'll see each other in 2 weeks' time, if I'm not mistaken. So everyone have a very nice afternoon and thank you all. Cheers.
Thank you very much. Thank you very much for your time and attention. I'm really glad that I could share our results with you. Thank you.
Thanks.
Thanks.